All right. So, yes, I am gone today, but I'm still going to help you walk through this quick little information about diffusion. So, we last left off with discussing the structure of the cell membrane. And so, remember, we have that main phospholipid bilayer, the polar heads, nonpolar, hydrophobic region in the middle, and all the helpful proteins that help do other stuff. Um, so, how can molecules pass through the membrane? Well, you need to be a couple of different characteristics in order to fit through and chemically go through that region of the membrane, the nonpolar part. So one thing you can be is small enough. So if you're small enough, you can kind of eke through the cracks of the membrane. So things that diffuse fast through the membrane are oxygen, carbon dioxide, and water because they're really small. They can fit through all the little phospholipids. Glucose can fit somewhat through the phospholipid bilayer, but since it's a little bit bigger, it's a slow diffusion. And some things can't diffuse at all through the membrane on their own. Things like proteins, which are huge, and long chains of sugar like polysaccharides. So, depending on how big you are, can determine how quickly you naturally diffuse through the membrane. You want to be nonpolar because the main barrier is the phospholipid tails. So if you're nonpolar enough, you might be able to go through the membrane. So O2 is nonpolar because it's two equally electronegative oxygens. CO2 it has polar bonds, but because it's symmetrical, it's not a polar molecule. But steroids and lipids, which you actually can get pretty big, can actually fit through the membrane because it's fine going through the nonpolar region. It's nonpolar itself. Ions, which are very polar, hydrophilic, do not fit through the membrane very well at all. Because remember, they're surrounded by those hydration sphere where the waters surround it and it makes it really bulky and polar. So they do not go through on their own through the membrane very well. If you're concentrated enough, you can kind of force your way through the membrane. The one example of this is mainly water. So water is really polar, but there's so much on the outside that it can actually force its way in and out depending upon which has the much higher concentration of water. And so finally, if you're like, well, how do I get those things through the membrane? You can have transport proteins. And so they're often tube-shaped devices that have different properties that allow the specific molecules to get through. So maybe it's just big enough for only a sodium ion, but not a potassium ion to fit through. And it has lots of negative inside so that only positive things can go through. So based on those R groups, different proteins will transport different things. And finally, any combination of those can equal allowing things to get through the membrane. So water will go slowly through the membrane, even though it's polar, because it's small and it's concentrated, can go through the membrane. But let's say you want a lot of water. Well, we have special proteins called aquaporins that allow a lot of water in and out depending upon the situation. And so you can have a protein as like a backup in case you needed to move a lot of water quickly. So water kind of fits almost in all these examples other than, B. So depending if you're small, if you're nonpolar, if you're concentrated, or if you have a protein, that helps you get through the membrane much easily. So here's kind of a quick check. Which of the following molecules do you think will diffuse the most quickly through a lipid bilayer membrane? So think about the properties and size of these molecules and take a guess. Hopefully you said oxygen. It's the smallest one there, and, well, maybe the sodium ion is smaller, but it's nonpolar, whereas the ion would be surrounded by the hydration sphere. So BO2 would be the fastest. So take a look at these ones, the correct order of molecules that will move from fastest to slowest. So we have three different ones. Who's going to be the fastest? And you should have gone with C because amylose is a polysaccharide. It'd be much slower and probably not much move through the membrane much at all. Anyways. Okay, so we talked about what properties allow things to go through the membrane more easily or not. But like, why do they move through the membrane? I mean, again, we talk about how we don't want to personify chemicals and make them think or do things. Well, the energy behind making things move through the membrane is kinetic energy. So All molecules that are above 0 Kelvin temperature has an inherent amount of energy in it, and then energy causes the molecules to vibrate back and forth. Okay, now this movement's random, they're just moving randomly. And so when you're imagining this, you need to imagine as if there are, let's say you're sitting in this classroom and there's 100 blind individuals in this room, and they're like we blindfold 100 students, and so they're just wandering around back and forth in the room. They don't know where they're going. And let's say I open up a door, and so eventually some of these blind students are going to wander out of the classroom. Okay. Now, when one person wanders out, it's going to be probably a while before they wander back in, because there's only one of them out here and there's 99 left in the classroom. So more of them are going to keep wandering out. So this random movement depends on how concentrated you are. The more people or molecules you have on one side, the more randomness might allow them to get through the membrane. So the key is that diffusion is a natural process. There's no extra membrane, but molecules are going to move from a high concentration to a low concentration. So if you think about it, if there's more molecules on one side randomly moving, they're likely to move to the other side. And eventually you'll reach what's called an equilibrium, where there's about 50-50 on each side, and so as one moves over to one side, another one moves back to the other side, and you're really not moving large amounts. So what is diffusion? Diffusion follows a concentration gradient. So you know, like in word art where you have like that color gradient where it goes from like one color to another. So the gradient's the idea that there's a high side, like a darker side, and a lighter side, which is your lower side. And so it's always going to follow from high to low. It does not require additional energy. We're not using ATP. We're not using sugar. The natural energy from the kinetic energy of the molecules is what's causing them to move. We call this passive transport because you're not actually doing anything. If you're a passive person, you just kind of let things happen to you. And so the molecules are passively going to move from one side to the other. And how a cell uses diffusion is one of the big things is gas exchange. So oxygen we just talked about a few minutes ago is very small and can move through the membrane no problem. So there's a lot of oxygen out in the environment. So you can see all these circles out here. And there's not a whole lot in the middle. And so these oxygens are going to randomly wiggle around, and some of them are going to go into the cell through the cell membrane. Then through the mitochondria, they're going to take that O2, take some sugar and burn it and turn it into carbon dioxide. And so now we're going to build up a ton of carbon dioxide in our cell, whereas in the atmosphere there's going to be much less. Or in the case of higher organisms, your bloodstream will have less. And so then after cellular respiration, we make all those carbon dioxide, they're going to diffuse out because, again, there's higher on the inside, lower on the outside. And so just through random kinetic movement, on average, you're going to have more leaving than entering. And so that is how we move gases without even trying, just through naturally having one side having more and the other side having less. Molecules will move as long as they can fit through the membrane via diffusion. So take a look at this picture. Which way will the solute, the orange circles, move during diffusion? And I will let you know that the orange circles can fit through the membrane. So you should have gotten moved to the right. If it wasn't permeable to the membrane, then they wouldn't move at all because they'd be stuck where they are. But because they can move, they're going to go from the high concentration to the low concentration. Because again, there's more on the left. It's going to balance out and move to the right randomly. Okay, so that's all I have for that. Your lab data should be coming up pretty soon for your dialysis tubing, so take a look at that. Try and figure out what's moving based on your results. Think about the semi-permeable membrane nature of your dialysis bag. Fill that lab in, and then you have some extra time to work on other stuff, whether that be your reading guide or your lab corrections. If you have any issues, you can hit me with an email. So I will see you guys on Thursday. Thank you.